Hello, and welcome to everyone joining us for today's Select Science webinar titled New Trends and Innovations in Automated Cell Counting. My name is Mathilde Marks. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speaker, Karine Labour, Europe Managing Director at Logos Biosystems. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of auditorium and click to open the Q&A window. During the presentation today, we will also be running a few poll questions. Please keep an eye on the engagement window on the right-hand side for poll icon. If you could please submit an answer when prompted, it would be greatly appreciated. So, without further delay, I'm delighted to hand over to Karine. I would like to thank her for presenting to us today.
Hello everyone, welcome. My name is Karine Labour. I am Europe Managing Director at Align Genetics. Thank you for attending this webinar on cell counting trends, challenges, and solutions we can offer. If you have any question, feel free to ask it during this presentation through the chat or contact us by email or through the website. We'll be glad to answer any request. First, let me tell you a few words about Align Genetics. Align Genetics was set up back in 2008 with headquarters located south of Seoul with two subsidiaries, one in the US and one in France for Europe. We collaborate with about 130 distributors worldwide, covering 115 countries. Our company is highly focused on innovation since the beginning, and as of today, we have two brands, GeneScale, which is an in vitro diagnostic point-of-care solution, and Logos Biosystems. Logos Biosystems is well known with its range of multi-awarded cell counters, as well as innovative products such as digital imaging systems and tissue clearing solutions. Today, I'll emphasize on cell counters and related questions you may have through examples such as cell lines, T cells, and isolated nuclei. We all know that cell counting is critical, but necessary. It's a necessary step that requires accurate and reproducible approach, whether you want to normalize experiments, enable decisions, or optimize downstream applications. This essential method offers numerous options from hemocytometer, such as Malassez or Neubauer, as you can see here, up to recent image-based cell counters like our Luna cell counters. You can see the difference already in terms of images. But it starts to integrate artificial intelligence, especially with machine learning approaches. At the end of this presentation, I'll show you how we include such evolution with our newest cell counter. The process of cell counting starts with your cells of interest, and you'll have different needs and expectations, leading to a list of challenges, or I could say priorities, to consider that help defining a short list of available solutions. You can therefore make an educated choice and select the best cell counter for your needs. But start with the cells, because the cells can be really diverse, from cell lines to primary cells or even bacteria and so on. If you are involved in fundamental research with cell lines, or if you produce products in your facilities with cell lines, your needs will be different. Regularly, you can face the presence of debris or contaminants, such like red blood cells, if working with PBMC or splenocytes, for example. Also, in the same sample, you can have cells with different sizes, and that's the case for MSC cells. Or you will encounter clusters or cells with different shapes in the same sample. And maybe you're interested in counting other kind of cells, less classical cells like pollen, protoplast, spores, sperm, ovocytes, microalgae, parasites, and so on. And if you're working with bacteria, extra challenges will have to be addressed, including genome size and Brownian movements. So lots of challenges to address just from the cells you're working with. And that's the first step. And when do you count your cells? Well, counting cells is a common procedure in scientific experiments. When you monitor the health of your cells, you want to check the proliferation rate, you want to assess immortalization, for example. You want to check the transfection or the infection, or at any time, you want to seed cells for subsequent experiments, especially cell-based assays. So it's critical that your cells are counted accurately with a reproducible approach, a robust approach, especially when you want to quantify measurements of cellular responses, but not only. Accurate counting is providing quantitative and qualitative information, such as concentration, viability, cell size, cluster, etc. Accurate counting will enable decisions from the passage to cell therapy development. During bioprocessing, living cells are used to produce product of interest. It can be an enzyme, can be a monoclonal antibody, and optimizing this process efficiency is key. Finally, accurate counting aims to limit inter-operator variability. Most of applications require a total variability lower than 15%, sometimes even 10%. So every step is a source of variability, a variability you want to limit, to optimize in order to improve experiments and processes. Indeed, the needs will differ between people, between labs. Ranking expectations is important. Are you looking for a flexible solution with the possibility of staining cells with different dyes, for example, in bright field and fluorescence? How important is accuracy for you, even if I do believe it should be critical for anyone? Are you counting few cells per day or hundreds? How many operators will be involved in routine? Do you have to generate reports? Is data traceability mandatory in your process? Is vendor support and quality control very important to ensure your trust? What's your budget for investment, for running cost? So many questions. So ranking, defining your needs and expectations will be extremely useful for the next steps. So once your expectations are clearly established, then assessing key parameters of available technologies will be essential. Depending on defined priorities related to your working environment, you may want to assess the accuracy and precision, the reproducibility of the cell counters, the intra and inter-devices viability, the throughput, the data generated, the support, the compliance options, the sustainable and eco-friendly options, compare the cost per count, and so on. And that's not easy, as there are different technologies and multiple technologies that exist to count cells. So here is an overview of the most important and well-known technologies. Using an hemocyter and under a microscope is the most inexpensive and well-known in the market, but we all know it's a tedious and time-consuming method with subjective results due to the perception of which cells to count or not, volume issues, pipetting errors, and inter-operator differences between two colleagues counting the same cells. You will have massive difference from time to time, and you know it. Cytometry is another option. It brings you far more information than concentration and variability, but it's quite expensive and time-consuming as well, and not everyone has access to a cytometer. Coulter method is an interesting method for particle counting, but to me less interesting for cell counting. This method is based on impedance measurements. It measures the changes in electrical conductance as the particles pass through a small orifice. When it goes to cell counting, a major pitfall of that technology is the price. You can go up to $4 per count, which is far too expensive for me. And there is a lack of control. You don't see your cells, and it's not efficient when it comes to samples contaminated with debris or contaminants. Image-based cell counters are the most common and affordable solution to count cells with variability dyes. Indeed, all image-based cell counters don't compare, and going back to your list of priorities is essential to identify the right technology. For example, we have 6 cell counters at Logos Biosystems. 6. You can imagine we propose multiple solutions since a single instrument doesn't match all expectations. So how to define, how to choose the accurate instruments and face all the challenges of choosing. Through 4 examples for different applications, I'm going to illustrate how and why our customers have chosen one of our cell counters. Applications I'll cover today are the following. Nuclei counting and size assessment with the Luna FX7, T-cell counting in cell therapy application, CAR-T, with the Luna FX7 as well, antibody production from cell lines and hybridoma with the Luna FX7, and fundamental research, counting of cell lines in fundamental research with the Luna 3. So let's start with isolated nuclei. And for example, in single cell experiments. For information, our colleague Joe made a great webinar recently about this application. Feel free to watch the replay because there are several tips and tricks inside this webinar. So cell counting analysis and nuclei counting is a method with incredible potential but quite expensive. Our customers highlighted that key parameters when choosing a cell counter from that application are the counting method, the accuracy and precision, the data, and especially the data consistency, and the cost efficiency. So let's go back to the process. So from analysis of rare cells, cellular map networks, characterization of abnormal cell populations, and so on. This technology leads to significant time and resources to reach high-quality data. All single-cell sequencing protocols rely on accurately quantifying cells and isolating nuclei before processing, meaning accurate cell counting is a critical, a vital step in this process. Nuclei isolation is crucial to enhance accuracy, leading to more reliable outcomes. Concerning nuclei counting, with the Luna FX7, you can either count in bright field with variability dyes such as trypan blue or fluorescence. Trypan blue is highly toxic to cells and impermeable. So it means it will not enter the cells if the membrane is intact, only when the membrane is damaged or removed. Trypan blue binds to proteins and nuclei, staining dead cells or nuclei in blue, which can be visualized under bright field microscopy. In fluorescence, Acridine orange is a membrane permeable that enters live and dead cells and binds to nucleic acids, emitting green fluorescence when bound to DNA. Propylium iodide, PI, or another alternative, which could be ethidium hemodimer 1, on the other hand, cannot go through the same membranes when the membrane is intact, and it will specifically bind to nuclei. So when the membrane is damaged or when the membrane is removed, so at the nucleus level. Both PI and ethidium homodimer emit red fluorescence upon binding to DNA. So the mix is AOPI or AO ethidium homodimer are commonly used to distinguish between live and dead cells based on their permeability and quenching effect, where the PI or the ethidium absorb the acridine orange signal. Basically, it will quench the acridine orange, preventing bleed through of double positive results. The signal of PI is basically increased by 20 to 30-fold at a time, leading to avoid double positive results. So therefore, staining nuclei or dead cells are red. We do not recommend trypan blue for nuclei counting due to several reasons, even if it's possible with the LUNA FX7. The nuclei isolation process can introduce debris or aggregation, which may affect the accuracy of staining results overall. Trypan blue staining may non-specifically bind to various particles, which can cause inaccurate data analysis. Furthermore, nuclei typically have relatively dimmer edges, which can make harder to distinguish the edges. This can also lead to inaccurate data analysis. Therefore, we recommend using fluorescence dyes such as AOPI, which can solve those issues. Let me show you how. When using fluorescence imaging, several factors will be considered when assessing the quality of the nuclei. So the green cycle here indicates intact live cells, which should be disregarded when assessing nuclei. Okay, you don't want to count live cells, you want to count nuclei. The red cycle highlights dead cells or nuclei, and it's crucial to observe the overall morphology, checking if the cytoplasm is completely removed from nuclei. As you can see here, it's not the case. Okay. Successfully isolated nuclei typically exhibit round and elliptical shape, while damaged nuclei may appear irregular. Debris is not a concern, it's not a problem, because it won't be stained, it won't emit a fluorescent signal, meaning any debris won't be tagged as objects. High-resolution images obtained with the LUNA FX7 can then be used to visually verify the quality of isolated nuclei. Data are therefore accurate and reproducible with the right method. Here, fluorescence with nucleic acid dyes. Another quality control step during this process is the cell and nuclei sizes. The LUNA FX7 can accurately discriminate from samples with large cytoplasm and small nuclei, such as HeLa cells, and cells with large nuclei relative to the cytoplasm, like the egg 293. So you can see here, for example, the HeLa cells. The cell size is 13.4 micrometer as an average, and the isolated nuclei is only 6.4 micrometer, so big difference. But when you work with the egg, the difference between the intact cells and the isolated nuclei is far less important. It goes from 13.3 micrometer down to 11.7 micrometer. So having extremely precise instruments to quantify the cell size when working with nuclei is really important. The LUNA-FX7 is proven to be around 98% accurate with cell size assessment when compared to NIST standards, so it's highly reliable. So as a conclusion for this application, the LUNA-FX7 is a nice tool for quality control criteria to determine if the samples pass or fail before committing on expensive downstream processing. A single experiment in single cell can cost you several thousands of dollars. So it's really important to make sure that your cell concentration and nuclei isolation is performed efficiently. The second application I would like to talk about today is cell therapy with the CAR-T example. You probably know the CAR-T cell therapy. It's a living drug established from patients' own isolated and engineered T cells. Production of autologous or allogenic CAR-T therapies is a complex biomanufacturing process that requires strict adherence to regulatory and quality guidelines. To ensure the safety, the quality, and the efficacy of the final product, the cell health and viability must be evaluated and monitored throughout the process of development and bioprocessing workflow. So as you can see, our customers highlighted that the key parameters for a cell counter when working in cell therapy is the counting method, accuracy, precision, and CV of the instrument, QC, compliance, support, and data quality and traceability. Briefly, to go through this process, there are 5 major steps in CAR-T cell therapy. At every step, robust, highly accurate cell counting must be performed. In the first step, you collect whole blood from the patient. Therefore, fluorescent cell counting is the only solution to count the cells of interest, which are the white blood cells. After that, you're going to remove the red blood cells and isolate the white blood cells. And at the time, also, you could have still contamination with red blood cells. So it's important to stay with fluorescence. Then you're going to isolate the T cells and activate it. And then transfect the chimeric antigen receptors, so the CARs, to create the CAR-T cells. Then make it proliferate, so you can keep on following up the concentration and the proliferation of your cells. Test it, condition it, and then infuse it, so inject it back to the patient. And even then, on that step, it's critical to have an optimal, a very accurate estimation of the concentration and the viability of the CAR-T cells before to ensure the success of the therapy. All those results can be found in a dedicated application note. We collaborated with Curacell, which is a leading company developing innovative anti-cancer immunotherapy. And all of that shows that the LUNA-FX7 may be used to monitor and evaluate cell health at critical stages of the CAR-T therapy. And this can only be reached because of unmatched accuracy and precision of the LUNA-FX7. As you know, accuracy is the proximity of results to the true value. You have a target and you want to be close to that target. For example, for the dose, for the infusion, you want to be at 500,000 cells per ml, then you can rely on the accuracy of the instrument. The precision which is also called reproducibility. The precision is the degree to which replicates show the same results. It's basically the CV of the instrument. And you can see here that the Luna FX7 has an intra-device CV of less than 1%, and that the inter-devices CV is below 2%. So it means that if you're collaborating, you have a Luna FX7 and you're collaborating with another lab, institute, or company, which is in another country, well, you know that the variability between the instruments would be less than 2%. So you can really more easily reproduce the experiments. And this is unbeatable. No other, or hardly unbeatable, no other affordable solution in the market, no other cell counter will be that accurate and precise instrument. And those CVs are validated for 3 parameters: concentration, viability, and cell size. A dedicated application note is available to illustrate it. So highly reliable results can be obtained whatever the slides you're using, because we offer multiple options in terms of consumables with the Luna FX7. As you can see here, you can have a 1-channel slide, 2, 3, 8-channel slides, and even a reusable slide. So for most of the slides, you're going to use 10 microliter per chamber, except for the 1-chamber slide where you're using 50 microliter. And within this volume, a significant quantity will be used for the counting. So you can see that even with the 8-channel slides, the minimum recommended by the guidelines of NIST and ISO international standard is used, okay, 0.5 microliter. And about almost 3 times more cells are counted with a 3-channel slide. So you can have the choice between consumables with this instrument. Of course, the Luna FX7 has a wide concentration range, let's say between 10,000 cells per ml and 25 million cells per ml, with some differences depending on slides. So going back to the cell therapy application, something that is extremely important for customers, for users, is the quality control, support, and compliance. Those are key parameters for this application. Supporting with IQ, OQ, PQ, preventive maintenance, and 21 CFR Part 11 compliance is essential. Quality of customer support is a top priority inside Logos Biosystems, so we are here for you at any time if you decide to adopt the Luna FX7 for your application. Nevertheless, the validation slides, we have developed validation slides. You can see here we have the fluorescence validation slide and the bright field validation slides. Those are kind of absolute standards. The validation slides are available to assess by yourself, the performances of the Luna FX7 over time in the lab, so in between 2 maintenances, for example. For GMP or 21 CFR Part 11 compliant lab, data quality, electronic data integrity, and traceability are at the heart of the process. The countwire, this is the name of the option we propose, has been developed to make the Luna FX7 compliant. So when we talk about compliance, we talk about electronic data integrity. So it means tracking everything. You can't modify your results. You can't delete anything. You will trace the people who are authorized to use the cell counters, which protocol they will be allowed to use. The validation of the results by managers and supervisors, the traceability of the data, the backups, and even the audits, the audit trail. Every time you do something, you need to have a reliable proof showing that those are the original data, unmodified data. Talking about data, data generated at each count can be saved on the Luna FX7. There is an onboard storage of either 250 gigabyte or 1 terabyte, or remotely on the server with when using the count wire. Onboard storage includes images, so raw images for each field of view, and analyzed images as well, PDF report and CSV file. And of course, complete analysis of your cells is saved with concentration, percentage of viability, cell size, cluster analysis, etc. You may have noticed that the parameters to assess the priorities are different depending on applications. The parameters that are critical for people looking at nuclei quality are not exactly the same than people working in cell therapy. But the Luna FX7, the same instrument, can fulfill each requirement. So after 2 examples of cells that require fluorescence counting, I'd like to show you some applications with cell lines in bright field. Cell lines usually don't need fluorescence to be counted accurately. So let's move on and let me show you some examples, some results with monoclonal antibody production. During bioprocessing or during antibody production, any production, different cells can be considered. And here, for example, the CHO cells are used by almost half of the companies, a bit more than half of the companies or labs producing monoclonal antibodies. Those cells are easy to use. They are well known, but they also have some disadvantages, like the presence of debris, especially at the end of the production. You expect to keep the same technology during the entire process, from preclinical to then clinical steps. You want to make sure that everything goes well, and as you're going to test thousands of compounds for only a few successes and hopefully bring it to commercialization after the authorization from the FDA or the EMA in Europe. So here are other parameters that are more critical, such as throughput and safety. In this process, bright field with either trypan blue or erythrosin B is recommended. Even more erythrosin B. Erythrosin B is a safe alternative to trypan blue, and a significant number of companies are avoiding trypan blue and request a safe solution. And as you can see here, counting quality of CHO and transfected CHO counted with erythrosin B in bright field with the Luna FX7 is are just perfect results. So erythrosin B is a safe alternative, and with the FX7, you have the option to remove a toxic compound from your process and adopt the erythrosin B instead of trypan blue without having to calibrate the instrument, because of a very nice feature we have, which is called auto exposure, and that allows you to switch from one to another without any calibration. And the good news is that it works also perfectly with myeloma and hybridoma. As you know, antibody-producing B cells are harvested from the mouse and fused with immortal B cell cancer cells, myeloma, to produce an hybrid cell line called an hybridoma, which has both the antibody-producing ability of the B cell and the longevity and the reproducibility of the myeloma. The hybridomas can be grown in culture, each culture starting with one viable cell, one viable hybridoma cell, producing cultures, each of which consists of genetically identical hybridomas, which produce one antibody per culture, so monoclonal antibody, rather than mixtures of different antibodies, so polyclonal. Production of different batches can be efficiently performed and tracked with medium throughput slides, either, for example, the 8-channel slide or the 3-channel slides. And for that specific application, we even have a software option. It's called the bioprocess software for the Luna FX7, and you can track batches, so the growth rate, the doubling time, and of course the concentration and variability can be followed. So you will have the raw data and graph that will be generated. Sometimes counting cell lines doesn't require throughput, neither the advanced possibilities offered by the Luna FX7. And in that case, the Luna 3 could be an interesting solution for you if you're in this situation. So this is our newest instrument, the Luna 3. The Luna 3 has multiple new features that I won't explain in details today, but if I got to summarize the Luna 3, I would say it's one of the most affordable instruments in the market, but with advanced algorithms trained by machine learning, so artificial intelligence. And there is even one package, including a reusable slide and erythrosin B, which makes the Luna 3 an eco-friendly and sustainable cell counter powered by machine learning. So when counting cell lines and doing fundamental research, what you want is to keep on having accuracy and precision, and but also have something which is affordable when you invest in this instrument, but also the running cost that is easy to use. And the Luna 3 is extremely intuitive to use. I mean, there are only a few buttons to press. We have some safe options, and this new generation is including for the first time some artificial intelligence. So talking about machine learning, let me explain you how it's been integrated into the Luna 3 with a short video. One key feature of the Luna 3 is a new autofocus mechanical system. It is a highly precise stepper motor adapted from the Luna FX7. This is a reliable, robust autofocus technology. This new hardware part is combined with advanced algorithms for cell recognitions. Those advanced algorithms are trained algorithms trained by machine learning. This is made to minimize any focus deviation and accurate cell counting. To make it clear, it's extremely robust to find cells and related focal plane, and this is powered by machine learning algorithms. The other algorithms for the counting were already extremely robust, but this is for the first time. For the first time, a cell counter of our range includes some artificial intelligence, and to me, it's probably just the beginning. So if you wish to know more about what other customers are thinking about our Luna counters, I mean, hundreds of testimonials are available on our website, but also on Select Science. You can see here there are more than 700 reviews. Our Luna counters have been awarded multiple times over the last years, and we are deeply honored to receive such positive feedback and recognition from customers and third parties. I didn't cover in details the specificities of all our cell counters, neither the other products, so I invite you to contact us for more information, or you can also find quite a lot of information from our website. So thank you very much for attending this webinar. I hope it was interesting for you, and I'm available to answer any question you may have. Thank you.
Thank you, Karine, for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We will now move on to the last part of today's webinar, the Q&A session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of auditorium and click to open the Q&A window. Please also keep an eye on the engagement window on the right-hand side for a pulse icon. If you could please submit an answer when prompted, it would be greatly appreciated. So, to kick off this final portion of today's event, the first question I have for Karine is, Karine, can these cell counters accommodate a broad range of diverse cell types?
Thank you, Mathilde. Yes, we have quite a versatile range of cell counters. Sorry, it was just an overview today. I didn't enter all the details, but our counters can count from cell lines, primary cells, PBMC, yeast, hepatocytes, bacteria, isolated nuclei, pollen, protoplast, and so on. So yes, a very diverse range of cell types is possible with our cell counters.
Thank you, Karine. And are these cell counters able to differentiate between live and dead cells if clustering is an issue?
Well, that's quite a good question, actually. Yeah, as soon as you're using the accurate dye, you can differentiate live and dead cells. But I agree that when there is a cluster, it can be challenging for some technologies. Fortunately, our cell counters allow that. I have to say that from day one, our counters have been recognized for having outstanding declustering capabilities, even from the first one, which has been discontinued, was Red Luna. And the Luna FX7, which is one of the latest instruments, it has such a high quality imaging component so that we can even differentiate dead cells in a cluster. So yes, we have quite outstanding capabilities of differentiating live and dead cells even in clusters.
Lovely, thank you. And do I have to count 8 samples at once with the multi-channel slide?
Oh, no, you don't. You can count up to 8 samples at a time, meaning from 1 to 8. You decide.
Lovely, thank you. Before the next question, I would just like to um, tell people that there's another poll question being launched any second. So if you could please submit an answer when prompted, it would be greatly appreciated. So the next question, Karine, is Luna FX7 an open system that has the flexibility to select the type and size of cells you want to count?
Well, that's a good question. Yes, you can differentiate cells by size with the software of the Luna FX7. And after the counting, you have a file that is automatically created. It's a CSV file, and you will have access to extra information that will probably help you to answer the questions you want to address.
Thank you. And is there storage on the Luna 3?
Yes, the Luna 3 has onboard storage. It, it, the size of storage is about 10 gigabytes, so it means around 400 counts that can be saved on the instrument.
Perfect, thank you. The next question is, can I still use Trypan Blue with the Luna 3?
Yes, of course. You can choose counting your cells with trypan blue or erythrosin B. You can choose both options. The Luna 3 is adjusting the exposure and the autofocus. So you can decide to look at the variability with trypan blue, yes, or with another dye like the safe alternative, the erythrosin B. It's possible, absolutely.
Perfect, thank you. And I can count yeasts. Which instrument is recommended?
Yes, for yeast, we have several devices that can count yeast, but the Luna FX7 is really superior to count yeast, even with very difficult ones, like, for example, Candida yeast. You can count yeast in bright field with methylene blue or fluorescence with different dyes such AOPI, FDAPI, and we even have other alternatives for very difficult yeast. So yes, absolutely, the FX7 is the recommended instrument for that.
Amazing. Thank you. The next question is, can accuracy vary due to the size of cell?
The accuracy of the instrument will not vary due to the size of the cells. We have a broad range of cell size. It can count from 1 micrometer up to 90 micrometer. And the accuracy of the instruments and also the reproducibility have been validated through the entire range.
Lovely. And which device would you recommend for cancer cells count?
Well, I would say it depends on the throughput you will have. If you're counting cancer cell lines, then I would suggest you look at the Luna 3 or even the Luna 2 if you have a lower budget. And let's say if you have a very high throughput or if you want to have more capabilities and if you count other cells like primary cells, for example, then the Luna FX7 will probably be recommended.
Perfect. Thank you. I think we still have time for another question. Um, in our lab, we are using Luna 2. What I have observed while using the cell counter is that I have used same twice and the results were different. Could you please explain the variability I am facing?
Hmm. Well, the first recommendation I would give you is to save the images and send it either to us or to your local distributors. Because there could be multiple explanations to explain why you have different counts. But my recommendation is when you want to test the same sample is to load twice on two different chambers, not count again the same chamber again and again, because you have a parameter which is called evaporation, which can make your cells move inside the chambers, so you won't see the same after, let's say, a few minutes, which is normal with any cell counter. So, but save your images and send it to us. We'll be more than happy to support you.
That was great. Thank you, Karine. Well, I'm afraid that's all we have time for today. I would like to thank Karine for today's discussion and presentation and for this lovely Q&A session. And thank you to everyone joining us online for submitting your questions. We hope that you have found this a worthwhile session. If we didn't quite get to your question today, we will follow up with answers to this after the webinar. And if you have any other questions after today's session, please feel free to email myself at editor@selectscience.net. If you are interested in receiving a certificate of attendance, this will be shared with everyone who has attended live today via email a few days after the event. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.